Assalamualaikum warahmatullahi wabarakatuh. Hello, the honorable committees, panelists, and all the participants of International Conference on Chemistry and Chemistry Education 2022. I'm Salma Fauzia from 11 Maret University. It is an honor for me to delegate my research team to deliver the research about anti-dandruff shampoo formulation from rambutan leaf extract or Nervalium lapaceum L as antifungal mycelia furfur. So, ladies and gentlemen, we know that shampoo is a product that is used to maintain hair. The most common hair problem is dandruff. The microorganism that cause of dandruff is mycelia furfur. Actually, the existence of mycelia furfur on the scalp is normal, but this fungus will grow quickly when the oil glands in the hair are excessive. Rambutan is a fruit plant that is widely distributed in Indonesia. Rambutan leaf extract contains secondary metabolites that can inhibit fungal growth. Alkaloids can inhibit the growth of fungi because fungi can grow well at pH of 3.8 until 5.6. Flavonoids and tannins can cause damage to cell membrane proteins, causing changes in membrane permeability and fungal cell membrane lysis. Saponins can break fungal cells, which can make fungal cells swell and grow. So that is why in this research we use the benefit of rambutan leaves as an extract for anti-dandruff shampoo. So for the materials and methods that we use in this research, for the tools, we have rotary evaporator, autoclave, and many other tools. And for the ingredients, we have rambutan leaves, distilled water, 96% of ethanol, and many other main ingredients. And for the methods, for the first one, we have making rambutan leaf extract, and then making shampoo formulation, and then making potato dextrose agar or PDA media, and then task mushroom rejuvenation. After that, McFarland standard manufacturing. And the sixth one is mushroom suspension manufacturing, and then task media creation, and then research and data collection, and the last one is data analysis. And for the result and discussion, here we have the first one is sample preparation. For the sample preparation, rambutan leaves were cleaned with running water and heated in an oven so that the rambutan leaves were sterile and dry from impurities before the leaves were used in the study. The process of refining rambutan leaves is carried out to increase the surface area of the leaves with a solvents during the extraction process and to make the withdrawal of the active substances in rambutan leaves easier. And rambutan leaf powder obtained was in the form of green powder with a mass of 251 grams. So this is the picture of the dry rambutan leaves from oven, and the right one is a rambutan leaves powder. And after the rambutan leaves becomes a powder, we did extraction. The extraction was done by putting the rambutan leaves powder into container and then soaking it in the ethanol solution with the volume of 2,150 milliliters, and then closing it and leaving it for the 3 days while stirring it every day. After 3 days, filter and soak sample with filter paper, and then next we evaporate the filter using a rotary evaporator until a thick and dark green extract is formed. And then after that, we did organoleptic test. For the organoleptic test, we see that clear shampoo formulation with 0% of rambutan leaves. And for the formulation of 10%, 15%, and 30% rambutan leaf extract was dark green because it matched to the color of the rambutan leaf extract. And the four shampoo formulations not roses due to the addition of pink rose fragrance in the shampoo formulation. While the thickness of the shampoo is very thick at 0% of extract and thick at 10% extract and liquid at 15 and 30% of extract. And then for the pH test, the results show that the pH of 10 of 0% extract shampoo formulation was 8, and the pH of the 10%, 15%, and 30% extract shampoo formulation was 7. According to Indonesian national standard, the shampoo has a pH of 5 until 9. Therefore, the first shampoo formulation in the study met the pH standard of anti-dandruff shampoo. And for the next one is antifungal activity test. So this is how we did a fungi cultivation in petri dish on the picture on the left side, and on the right side, this is how we test an inhibitory of each of on each variation of shampoo formula. The result of the antifungal activity of Manzanilla for shampoo and ginger from the leaf extract are shown in this table, in the table 1. So anti-ginger shampoo with concentration of 10%, 15%, and 30% as positive controls in each treatment show that there were clear areas around this paper called the zone of inhibition. The diameter of the inhibition zone was getting bigger as the concentration of rambutan leaf extract increased. The inhibition of zone with the largest diameter was found in shampoo with a concentration of 30% rambutan leaf extract, while the lowest diameter of the inhibition zone was found in shampoo with 10% concentrated rambutan leaf extract. Thus, the higher the concentration of rambutan leaf extract in the shampoo formula, the greater the antifungal activity of Maltesia furfur. And this is the picture of the result of the antifungal activity on the Maltesia furfur. So for the picture A is 0% of rambutan leaf extract, and the picture B is 10%, and the picture C is 15%, and the picture D is 30%. And then we did test of normality. In this table 2 shows the result of the normality test using the Kolmogorov, Smirnov, and Shapiro-Wilk test with a significant value of the concentration of rambutan leaf extract in shampoo over 0.05. So the data is normally distributed. In addition, the data from the study were tested for homogeneity. On the table 3, we can see the homogeneity of the test result with the significance value of over 0.05. So the data were homogeneously distributed. Furthermore, on the ANOVA, when the ANOVA test was carried out with 95% confidence level on this table 4, with the result that there was a significant effects of a significance value of 0 is less than 0.005. There was a significant difference between the concentration groups of rambutan leaf extract in anti-dandruff shampoo. So that's why the result of one-way ANOVA test show that the zero hypothesis was rejected. So the next step was to carry out further tests, which is post-hoc tests, to see which test group were different. And before we're going to the post-hoc result, this is how we have discussion. So there are 3 groups of antimicrobial inhibition based on the diameter of the inhibition zone. According to the Pan et al., stated that the antimicrobial inhibition group based on the diameter of the inhibition zone was determined as follows: an area with an inhibition zone diameter of 0 centimeter to 0.3 centimeter had a weak growth inhibition response, and for the 0.3 centimeter to 0.6 centimeter had a moderate growth inhibition response, and for the areas more than 0.5 centimeter have a strong growth inhibition response. Based on these categories, the effectiveness of the anti-dandruff shampoo of rambutan leaf extract with a concentration of 10%, 15%, and 30% concentration has a strong fungal growth inhibition response. And this is the result of post-hoc test. In this table, 5 indicates that there is a significant difference in antifungal activity between bases or 0% and F1 10%, F2 15%, and F3 30% because P less than 0.05. This means that there is a significant difference between the average diameter of the inhibition zone of the fungus Mycosia fulfur with a concentration of 10% and a concentration of 15% with a concentration of 30%. So from this research, we can conclude that the shampoo with a concentration of 30% rambutan leaf extract had the best effectiveness in inhibiting the Mycosia fulfur fungus with the largest inhibition zone diameter. And from the analysis data from one-way ANOVA test, we know that the significance number of zero is less than 0.005, so it can be concluded that the average of the three test samples has a significant effect. And then rambutan leaf extract shampoo with this three concentration was proven to give strong fungal inhibition result. Thank you for your attention. From me, it's enough. Wassalamualaikum warahmatullahi wabarakatuh.